Hi, my name is Dr. Ben Temperton. I'm a research scientist at the University of Exeter and I study microbiology, and I'm leading the Citizen Phage Library project.
So what is a phage?
So phages are naturally occurring predators of microbes. So they are these basically nanoscopic style predators that then combine to bacteria, inject the DNA into the bacteria and overtake those bacteria to then produce more phages. And in doing so, they kill that bacteria.
Fantastic. And what's phage therapy? Why is this useful?
So obviously, as phages are natural predators of bacteria, you can imagine how they might be useful to treat bacterial infections that are resistant to antibiotics. So when phage therapy was first discovered, it predated the discovery of antibiotics, and so they found initially that they could use phages that they'd isolated from the environment and actually treat bacterial infections in First World War troops, and then they thought, well, maybe we can use this to treat kind of standard bacterial infections. And then obviously antibiotics came along. That was seen as kind of the silver bullet way to treat bacterial infections. And so at least in the West, phage therapy kind of declined in its importance a little bit. But now with the kind of resistance coming back from antimicrobial resistance and all these antimicrobial resistance genes that are profligate in the environment, phage therapy is now kind of undergoing a resurgence in the West.
And what are the advantages of phage therapy over antibiotics other than the resistance issue?
So initially, one of the major advantages of phage therapy is firstly, phages are actually quite easy to identify. So you can go out to any environmental sample, you can probably find phages that infect a bacterial infection. Whereas for a standard sort of antibiotic, you're looking at maybe a 10-year pipeline and tens of millions of pounds to develop that into a drug. So phages are firstly cheap. And secondly, whereas your antibiotic is actually going to be targeting all the bacteria in your gut, so it will wipe out any of your kind of gut microbiome that we know is actually quite beneficial to human health, phages are highly specific. So they will bind to the surface of the pathogen itself and then kill that, but they will leave your kind of friendly bacteria behind.
Fantastic. So what's the potential impact of this Citizen Phage project?
So the main purpose of the Citizen Phage project and its primary impact is to basically build a UK capacity for phage therapy in the UK to meet the growing antimicrobial resistance threat that we're currently having. And we thought the best way to do that is actually to reach out to citizen scientists to take as many samples from as many different sites as they possibly can, and then we try and isolate phages from them so we can build up a very rapid library of phages that we can then use to treat these pathogens as they emerge.
What's the process then once somebody collects a sample?
So firstly, the citizens will go out and collect the samples with the sample kits that we send them. And then they send those kits back to us, and each sample jar will contain, say, 40-ish ml of fresh water. We then centrifuge those down to remove any bacterial and filter them to remove any bacterial particles. And then that leaves just the sample and any viruses that might be in that environment. And then let's say we're looking for a phage that infects a certain type of E. coli, we'll then add that E. coli to that sample. And then if there are any phages that infect that E. coli, they will then grow over time, they will lyse those cells, they will produce more phages, and then we can essentially take those phages out and purify those cultures and then sequence their genomes to make sure they're suitable for phage therapy. So one of the things that really excites me about this project is this idea that we are running out of tools to treat antimicrobial resistance. So they estimated by 2050 it's going to be an additional 10 million deaths worldwide. Phage therapy is a really promising tool to basically help us not only extend the life of our current antibiotics that we have, but also to create novel antimicrobials that we can then use. We can develop them cheaply using this particular method, and we can basically roll them out and specialize that treatment to that particular person. So it's the ultimate combination of kind of cheap, reliable, cool biology and personalized medicine that we can push forward in this method.
So how did you get where you are today? What was your path?
So my path, I started off as a software engineer many years ago. So I spent 7 years as a software engineer developing systems for the NHS. And then I basically had an early mid-career life crisis and decided that I really wanted to return to science. I'd previously done a chemistry degree and actually really enjoyed it. So I retrained as a marine biologist down in Plymouth University, and then largely fell into marine microbiology. So I spent a good number of years being a marine microbiologist and looking at how viruses influence global carbon cycles. Then in the last 18 months, we started working on sequencing COVID-19 because we just happened to use the technology that was being used for that. And that kind of got me introduced to kind of the clinicians and really started pointing out the real desperate need on the kind of, in the clinic for actually having novel antimicrobials. So through that, we kind of decided, well, actually, let's kind of redirect our research and start pushing into using our knowledge of phages to then find new drugs that can treat these things. So yeah, basically software engineer all the way through to marine microbiologist, and then now clinical microbiology is where we are.
So I'm here with Ben. We've got our Citizen Phage box, our kit, and we're going to go and try and see what we can find in the water. Okay, so where's the best place to go?
So basically anywhere with bacteria. So one of the reasons fresh water is really good for us is A, it's easy to process in the lab, but also this water is teeming with bacterial life. You can probably see there's kind of a scummy surface on there. It's actually going to have a lot of bacteria associated with it. So we'll just go around to somewhere hopefully where we're not going to fall in when we take our sample. And then we just basically want to take one of those jars and then fill it up with water, and then we'll send it back to the lab and see what we get.
Brilliant. So let's find a nice space. And probably worth saying, do take a friend in case you do fall in. Good to do it in pairs.
What kind of water samples would you be looking for?
So pretty much anything that we know is going to be bacterial life, which is almost everything. One of the rules we do have is ideally no pet waste or anything that is potentially going to have viruses in that may be able to infect people, just because that kind of reduces the risk of us having to kind of use extra special steps in the lab. So fresh water is great, any kind of seawater is great, riverine samples are amazing, kind of estuarine samples would also be really good. Bird baths are really good, you get lots of bird associated viruses, so those things are really excellent to use. So basically, yeah, anywhere you can safely collect a sample, we will take it and we will process it and see what we get. So. Okay.
Brilliant. So what have we got in our box?
Okay, so in the box you will have a pamphlet that will basically give you the instructions that we're about to go through, and then you should have 3 jars, each of which has a label on it, and we will use those to identify the place in which we're taking our sample from. So the place we'll find is on your mobile phone. If you have an app called What 3 Words, you can basically find on there the position you are in. So currently we are standing in a square called Quarrel Smashes Clap. So you can write those 3 words on the label.
Quarrel?
Quarrel Smashes Clap.
Which is clap. And then we've got a slot for the date here. Okay. Brill.
Okay.
Right.
So then you just need to unscrew it and then fill it up with water from there.
And how full do you need it to be?
I'd probably take it up to the neck. So it's normally about 40 ml will fit in that jar. So up to the neck is fine.
Brill. So we've got some fairly scummy water here. Whoa. And does it matter if it's got chunks in it?
No, not really. Those will be, when we process them, the first thing we do is kind of spin down any sort of debris in there. So it should be fine there. So that looks like a good sample. Yeah, so put the lid on very tightly.
Very tightly. Right.
And then you can put that back in the box.
Fantastic. I might want to wipe the edge first, I think. Brilliant. There we have.
Okay.
So hopefully there's the key to curing some superbugs in here.